one of the big leaps that we had to make is overcoming the uncertainty associated with receiving seed support from a non-traditional investor. I talked about how there's not really a blueprint, but there are some elements that are well defined in the protocol of taking technology into a startup. And one of the things that is almost treated as law is that you will take traditional
venture
funds, right?
The angels and the ventures, yeah.
Exactly. And we reached this turning point where we sort of had some term sheet options over here from traditional VC, and then we had this non-traditional investor. And we ended up going with a non-traditional investor. And a lot of people really would take pause at that decision. And we have a network of trusted advisors. And it was something we took very, very seriously, deviating from the norm in this way. At the time, it was kind of a leap of faith, and you never know how the landing is going to go.
Hundreds of thousands of researchers around the world are working to improve life and address imminent threats to humanity. Often their research ends up in the scientific valley of death in the form of publications and patents that never see the light of day. That is about to change. Welcome to the Lab to Startup podcast, hosted by Naresh Sunkara, founder and executive director of the Berkeley Postdoc Entrepreneurship Program at the University of California, Berkeley. This show has two main goals: share the stories of those who have successfully founded startups based on their own research, and highlight resources needed to help those aspiring to launch startups in the deep tech space. Whether it's electric cars, vaccines, addressing climate change concerns, or possibly establishing life on other planets, Naresh and his esteemed guests want to help scientists, engineers, faculty, and researchers bring their innovations to market. Learn more and subscribe today at labtostartup.com. And now, here's Naresh.
I have two very special guests today, Dana Foss and Ross Wilson, the co-founders of Editpep, a biotech startup focused on developing CRISPR-based therapeutics. They're using a proprietary peptide-based delivery platform that enables targeted delivery to specific cell types, particularly for hard-to-reach areas like the brain. Ross was a postdoc in Nobel Laureate Jennifer Doudna's lab, and now he has his own lab and collaborates with Jennifer Doudna at the IGI. In this episode of Lab to Startup, we will explore the initial decisions that led to the launch of the startup, existing CRISPR delivery technologies, their challenges, and then do a deep dive into their technology platform. And then we discuss the opportunities in the space, their fundraising efforts, and the future goals. I've been following Ross's journey for a while and also got an opportunity to know more about Dana recently. Delivery of genetic therapeutics is of great personal interest to me as I spent my postdoc research on this subject at UC Berkeley. I really believe there is a lot to learn from Dana and Ross about how they're building the startup. I hope you'll enjoy this and learn from this great conversation. All right, Dana and Ross, welcome to the show. Thank you for taking the time. I know you guys are very busy, but I've been wanting to do this for a long time. I'll start with Dana. I would like you to set the stage for the conversation by asking you about the problems you're solving at Editpep. Give us the lay of the land about challenges around CRISPR on the therapeutic side and the delivery front, especially with the existing solutions. And then we can dig into what you're doing.
Perfect. Thanks, Naresh. Yeah, it's so great to be here. Thank you so much for having us. Really excited to be on your podcast. We're fans. Yeah, so at Editpep, I'm CEO and co-founder of Editpep together with Ross Wilson, who's here today. And at Editpep, we are trying to solve the delivery problem for CRISPR therapeutics. So we're coming up with a new way to deliver CRISPR as actually a functionalized enzyme into human cells and tissues.
And what are the current solutions you have, and what are the challenges with that? Why do we need Editpep?
Yeah, so delivery for any macromolecule is actually really challenging. So that any sort of large molecule that you need to get into specific cells to have its therapeutic purpose inside the cell is a major challenge. So that's true for RNA silencing, siRNAs, so any sort of oligonucleotide-based therapeutic, any protein that has to have its effect inside the cell. So that's always been a major challenge for this entire sort of therapeutic area in genetic medicines. And we realized that although there's been huge advances with using viruses to get into cells as well as lipid nanoparticles, there's a lot of unmet need where those delivery technologies don't solve all of the problems in every tissue. And so we've had this idea that has remained relatively unexplored that Ross's lab is really dedicated to, and that is delivering CRISPR as a biologic. And so that fills an unmet need in the delivery technology space, especially because it has a small particle size and can basically get into places that nanoparticles can't, especially in the brain, and distribute well there. And it has much lower immunogenicity potential than a viral delivery agent. And so that's where we saw the currently available delivery technologies really, they were invented for other purposes in traditional gene therapy. And in terms of their success for delivering CRISPR, they're just not always the right fit. And that's because CRISPR is unique to exert its therapeutic purpose. It just sort of has to get into the cells of interest one time. You don't need a lot of it. And then you want those CRISPR enzymes to go away after. So it's very unique, actually, in how it exerts its therapeutic effect, as opposed to antibodies or siRNAs, things that you would need to redose a lot. And so it gives us the opportunity to be really creative and inventive. And so that's what we are aiming to do, is develop a delivery system that is really bespoke for CRISPR, but also sort of plug and play in that it can be used in different CRISPR modalities like base editing or different nucleases. So that's what we're up to at Editpep. And it's really all started in Ross's lab at UC Berkeley, because that's where I was training when we started Editpep.
Great. Thank you for that. And Ross, I'd like you to dig a bit deeper into the technology aspect. How does your delivery platform work, and what specific advantages does it offer over the delivery methods?
Yeah, Dana introduced things really, really nicely. One bit of context that I would like to add is that in the earliest days of people trying to turn CRISPR, the enzyme, into CRISPR, the technology, or CRISPR, the drug, it was a lot of geneticists. This is a natural type of person to pick up this new technology, of course. These are the people that knew what they wanted to change in the genome, and they really like having tools at their disposal. Typically, genetic engineering is mediated by some sort of plasmid, for example, or a lentivirus. So the first people to implement CRISPR are very comfortable with kind of programming the genetic code for their cargo. And this goes also for people with the background in traditional AAV vector gene therapy, right? There's this natural inclination to just program in the stuff that you want to have in the cell. And so this is why we got this first wave, and I think it still continues in academia, this first wave of virally vectored CRISPR delivery, in vivo in particular. This works really well to get you a Nature Biotechnology paper. It looks like all the problems are solved. But as soon as you start really thinking about what it looks like to run that whole marathon and reach the clinic and help patients in a way that is both safe and effective, a lot of problems start to emerge when you think about viral vectors. The long-term expression can increase off-target risk. The manufacture is a real headache. Even with a single dose, it's an incredible technical challenge. It slows down R&D. So, viral vectors are burdened with major issues. And I think there's also the widespread limitation on packaging size. It's really hard to fit a lot of cargoes into an AAV, for example. So although it may look like a lot of delivery challenges are solved, this is kind of limited to mice in a lot of cases. And we've seen a big sea change, I would say, in how people are trying to deliver CRISPR in the clinic. There's been a lot of pressure to shift towards what we broadly call non-viral delivery technologies. And that's for some of the reasons that I talked about. And so fortunately, people that want to encode CRISPR have access to lipid nanoparticles, which can encode CRISPR cargo as an mRNA. And this is a pretty useful technology, and there's a lot of promise in a lot of contexts. But as Dana highlighted, lipid nanoparticles have their own limitations. The particle size is, they're called nanoparticles, but sometimes they are kind of inching towards micron particles. In the brain, you really need a small particle that's kind of the size of an antibody, an IgG. And unfortunately, these CRISPR effectors, whether it's a nuclease or a base editor, they're about the same size as an IgG, an antibody. And so we've seen, and we've published on this, that you can really spread through brain tissue very nicely when you use a CRISPR enzyme in this ribonucleoprotein format. So that's a shorthand that I guess Dana and I will probably be using. RNP is the name for the preformed CRISPR enzyme, and it's active, ready to go state. So I guess I'll do a little proselytizing for the RNP. It's a really manufacturer-friendly format, and this was borne out in the first CRISPR drugs to make it to clinic. The ex vivo trials that happened pretty quickly rely on CRISPR enzyme in the RNP format, which is sent into these cells outside the body using electroporation. I think those trials proceeded really rapidly. We got our first FDA-approved CRISPR drug out of this, Casgevy, of course. And so that sort of told me RNP is a nice format to work with. And this kind of goes into a little bit of the common background that Dana and I have. We're not geneticists. We're both trained in structural biology, biochemistry of RNA, protein, and RNP complexes, and some biophysical background too. And so Dana and I really had this strong harmony in terms of how we look at different challenges. And I think we brought a distinct perspective to the delivery space because for us, it was a little bit less daunting to work with these enzymes in their enzyme state. It's something that had been left behind by most of the field. And my postdoctoral mentor, Jennifer Doudna, who's also a longtime collaborator, ongoing, she's really the first one to ask this question: can we take preformed CRISPR enzymes and put them in the brain and get editing? And the answer was yes. And it happened kind of in a way that seemed shockingly easy. So with that burst of invention, I've been working in my lab, strongly bolstered by Dana's work when she was a postdoc in my lab, to really push this CRISPR as a biologic as far as it can go for in vivo delivery. And so, yeah, we're excited about the progress we've made on that front.
Great. You bring up a very important point. I'll start with the first one, that it sometimes needs fresh eyes, outsiders, to come in and look at a technology from a different angle. I think that collaborative effort needs to happen in academia a lot more. I think we can get to solutions much faster. I'm glad you're doing this in this space. And the second thing it reminded me is that most startups die because of a lack of distribution. I think what you guys are doing to CRISPR technology is you're providing that distribution, carrying that payload to the site of action. You can change the game for good for a long time. Thank you for this. Maybe we can transition to the next step about your lab to startup. Dana, tell us about your journey from being a postdoc in Ross lab to founding Editpep. What inspired you to make that transition? And if there are any key challenges in that transitionary process that you had to overcome, that'll be great.
Yeah, absolutely. I think it's a different story for everyone that lands up starting a company out of academia. I think it's always quite unique to the particular circumstances of the technology, the funding environment at the time, the people involved, how many sort of folks on the academic side are involved. So, but preface just to say, every story is different, that's all. But with our story, what happened was, as a postdoc, I helped lead a highly collaborative project, actually, with Alex Marston's lab that was led by David Wen, as well as Justin Akam's lab at UCSF. And that allowed us to first test this new delivery technology in cells ex vivo, which is, cells in a dish. And that sort of laid the groundwork of the technology to be able to then be further developed in Ross's lab for in vivo delivery. And so what was really cool about the postdoc project was this collaborative nature. Even Ross's lab at the time, we were still quite small in Ross's lab, and we were kind of functioning like a startup, I would say, because we all had a shared goal. It was a little bit different than sort of every postdoc on like a different project. We were all really trying to come together and work on this shared goal of developing RNP as its own delivery modality. So that aspect, sort of managing a highly collaborative project with a shared goal, I would say gave a taste for what startup life could be. And I really liked that. And I really liked that we were all working together on the shared goal. And then when we had success enough to file a provisional patent, I would say that the wheels started churning with both Ross and I, and it became a conversation between Ross and I from the very beginning. What do we do with this technology? Do we find another company that wants it? Who would want it? It really became a very long brainstorming phase of like, who would benefit the most from this technology and how should we get it out there towards the clinic? And that was still when the technology was focused on ex vivo. And that is sort of letting people avoid electroporation for making cell therapies, because we have this technology that works both ex vivo and in vivo. And so Ross and I just spent a lot of time brainstorming, having a few early meetings with potential companies that would be interested. And everyone was really interested. But I think what became apparent to us is that it would be hard to shepherd the technology into industry without doing it ourselves to a large extent. Like, we wouldn't be able to find a company that necessarily was willing to try it on in this early phase, sort of launching it out of academia, do the process development to get it ready for industry. And so over time, it became apparent, like, okay, I guess we should do this and we should get it ready for clinical validation. And then assuming that along the way, we would also find larger partner companies sort of once we were more established. And so it was through all of that brainstorming and early conversations that it became apparent that we should really start this company. And Ross did a ton of the legwork because I also left my postdoc position for an industry job at that time. And so I worked at another startup in a pretty different therapeutic area, but also focused on delivery. And I got a taste for a couple of years of doing preclinical development with mRNA LNP therapeutics, as well as siRNAs and ASOs. And so that was really fascinating. And so I was basically having conversations with Ross the whole time around how we can get Editpep off the ground. But then Ross really took the reins in that phase too. And I'll let Ross take over on that part of the story, because he really took the reins at that point, because I had a full-time job and a new baby. And so Ross was leading the charge for a bit. So yeah, I'll turn it over to Ross to tell that sort of origin story at that phase.
This is a fascinating story. You bring up one important point about how when you're a postdoc in Ross's lab, you're operating almost like a startup. It reminds me of Steve Jobs. That's how he talks about how everybody in the company is working towards one goal. It's set up like a startup. Everybody's important for that final product, right? Ross, like, where did that thought come from? Like, you worked with Jennifer Doudna as a postdoc. Is that something you picked up there because Jennifer already launched startups, I guess, at that point when you were a postdoc? What influences did you have? And we can cover the rest of your story about how the complementary story to Dana.
Yeah, I think the road to startup was very natural for technology coming out of my lab, simply because of where my lab was situated and what our mission was. So I should comment a little bit that although my home base is UC Berkeley, we're also part of the Innovative Genomics Institute. So I have a role there where I am Director of Therapeutic Delivery, right? And the special thing about IGI, which is Jennifer's organization dedicated to maximizing the positive impact of CRISPR in the real world, the cool thing about IGI is that we really try to focus on that end of the marathon aspect that I talked about earlier. So when I started my lab, there was an explicit focus on coming up with technologies that would remove barriers to CRISPR therapeutics reaching the clinic. And we've been strategic in focusing on those real world hurdles that can kill a drug or prevent it from even advancing towards serious preclinical development. And with that mission, if we are at all successful, there should be some sort of entrepreneurship or startup momentum that's kind of naturally building, right? When you see real world problems and your lab seems to have a solution, it is not a question of if it's going to be translated. It's a question of how you're going to do that. And I will say that my lab has different pieces of technology that have taken different paths. So right now we have a collaboration ongoing in Brazil where we are working to use our ex vivo delivery technology to enable the first, potentially, CRISPR therapy for sickle cell disease in Brazil, where there are tens of thousands of patients, but no access to this transformative type of medicine. And that's an opportunity where we found a way to support that work that would not really find enthusiasm in the US because there were so many people trying to develop their ex vivo CRISPR therapies. And, we saw we had kind of one winner and then a bunch of runners up who have largely dropped their programs. And this is pretty natural for drug development, but that's one of those technologies that has real world utility, but only in the right context. So finding that match between the technology and the unmet need and then the right setting was really important. And that's why ultimately Editpep has taken a big focus with their internal pipelines on CRISPR therapy for the brain, because we found what we believe is a distinct solution that is looking great now. And as things build, we think we're really going to pull away from the other non-viral delivery technologies that have really struggled in the brain. So when I identified that as a clear match between unmet need, functional technology, and the right context, that sort of triggered this entire process and sort of kicked it into high gear. Because Dana was talking about the ex vivo applications in the early days. Those are still really valuable, still really interesting, and being explored by Editpep. But it's not the sort of thing that you could completely build your company around, right? So once we found this intersection and the harmony between all these elements for the brain delivery, that was when things really got serious and found that momentum.
That's great. I think what stands out is the intentionality. I think from the beginning, there was always a goal that we need to go towards helping people in terms of therapeutics, right? And Dana brought up a question about like, after you develop the technology, you ask yourself, what do I do with this? Should we license it to somebody else, or is it going to fall upon us? That resonates so much with me behind this whole podcast as well. Like, I think that question needs to be asked by every researcher in the lab. What do we do? Just publish and move on to something else? Like, it defeats the whole purpose, I think. Adding a little bit of intentionality and planning ahead of time of like, what are my goals going to be beyond a publication and getting a job, right? I think the world will be a much better place.
Yeah, and it's a hard phase for getting the technology out of academia. And luckily, we're in an environment in Berkeley in the Bay Area where there's quite a bit of infrastructure for getting technologies out of academia into startups, et cetera. But I would say it's still very daunting for an academic to know even where to begin, in who to ask these questions to, and where to learn all of the business side of things and how licensing works. And yeah, it's just a lot to teach yourself. So yeah, I think luckily Ross and I like talking to each other. And so we got to brainstorm a lot and sort of kick around ideas. And also Ross has a wonderful network of folks in the Bay Area that have started companies. And so, yeah, we are just sort of clawing our way through this landscape to try to understand how to translate this technology. But yeah, it takes a lot of self-motivation and a lot of, I guess, gumption to go out there and find out all the answers for how to take the technology to the next step.
Self-motivation and gumption, they're absolutely needed. Ross, yeah. Dana mentioned that you both like talking to each other a lot, which I've seen the chemistry earlier too. Can you share what those initial conversations look like as you launched Editpep? What I'm trying to tweak out of this is go or no go questions. Like, how did you come to conclusions? Like, what would those arguments look like? Any color to that will be great.
That's a great question. I don't think we've clashed very much. Dana and I have a lot of respect for one another. I can only speak for myself, but it helps that we have this kind of shared worldview, shared levels of optimism versus cautiousness. So we very much find ourselves in harmony. And I think it's been a really surprisingly easy journey working with Dana in this regard. And I think part of this is just luck. But yeah, I mean, there's certain conversations you can have with a potential partner or a potential investor where you do your pitch, and based on the way that is received, the message is kind of clear, right? So I think the earliest conversations were just taking one vision for the company, putting it in front of somebody who might be interested, and just gauging interest, right? And we kind of kept evolving what that vision looked like until we found a match, frankly. And that was a kind of slow and steady process. I think that other startups take a very different journey. We sort of took our time to craft things until it was a really nice fit. And part of this was like chance, there's been this downturn in biotech that everyone is aware of. So the downturn was happening right as we were crystallizing our vision. So fortunately, we didn't get swept away in the kind of pessimism. But it was a really interesting scenario because we had this vision, our pitch for Editpep, that we felt really good about. And looking at context, if we had been out there, 3 years prior with the beautiful in vivo brain data that we have, would have been a kind of instant green light. And so as our vision got stronger and stronger, money became harder and harder to get. So it was a very interesting luck-based journey in that regard. But yeah, unfortunately, I don't have any great stories about conflict resolution. And I think that's just due to some good luck. But yeah, Dana, if you had anything to add there, I welcome your input.
Yeah, we have a great working dynamic. And so there's great communication. So I think that helps a lot because there are a ton of decisions to make and also like a lot of dynamics to navigate when you're talking to potential funders or just anyone, all the sort of micro decisions that go into starting a company, it's great to have somebody that you're aligned with in more than just the science, right? Just sort of, as Ross said, sort of how you go about your work and how you approach social situations and how you approach all of these sort of interpersonal things. It really helps to have a co-founder that you're well aligned with. And the thing that was the most difficult at the beginning was this decision of whether to stick with the ex vivo technology or then to include the platform that had been further developed by another postdoc and others in Ross's lab since we had started the company. So there was sort of this transition period where we had one piece of intellectual property from my ex vivo work, and then there's another piece because the platform further evolved under the work mostly of Chris Baer in Ross's lab, as well as other graduate students and many collaborations. And there was many people involved. But that piece really, like Ross said, once that work really took off, we realized that this is really the thing that the company should be focused on for its internal pipeline. And so I wouldn't say there was conflict. We just really had to hash out a lot of aspects of that. And so that process can be challenging for a lot of people because a lot of decisions have to be made. But yeah, we were lucky, and maybe it's more than luck. I think we probably started working together because we have a shared way of seeing the world. And so, yeah, I think that really helps when you find a mentor, a postdoc mentor that you are aligned with on more than the science that can help this next phase. If you can imagine that you would like to start a company with that person, that goes a long way.
Yeah, I think that chemistry is really helpful. I'm very happy that you found each other in terms of like the co-founders, right? The chemistry is important. I see a lot of startups infighting and people falling apart, startups falling apart. I'm glad you crossed that bridge without having to fight with each other. Before I move on, Ross, I have a question for you. You still have an academic position at UC Berkeley. You're an adjunct assistant professor. I really think it's an innovative way you're building the startup. And I think it's a great model for other academics who are thinking. Can you shed some light on what's going on there and what lessons can we take in terms of encouraging other professors or postdocs from having similar titles to build companies?
Yeah, it's a great question. As Dana noted, there are many options to go from lab to startup when you're at UC Berkeley. And when I just started down that path, I would say it was almost overwhelming how many different sources of guidance and mentorship and assistance were available. For me, fortunately, I applied to Life Sciences Entrepreneurship. LSEC supported us to get into Skydeck. And it's a little bit interesting because Skydeck tends to be traditional technology, so software, but about 10% of the groups in my cohort were some sort of biology or biotechnology invention doing the journey via this accelerator program. That taught me that as a professor who wants to start a company, you can kind of dial up or dial down your investment, your commitment. The cup has to be filled up somehow. So if you're only bringing 50% of the contents, you have to find somebody else that's going to fill out the rest. There was some of the times when I felt like I was doing 75% or 80% of the work. And it can be challenging, maybe slows things down a little bit, but it's doable. So I guess for other people considering going on this journey, I think it's really important to evaluate and kind of continue reevaluating what are all the needs here, and do we have enough bandwidth and enough person power to make sure that the cup is filled. So I've been really fortunate in that with Dana coming on as a full-time CEO, this is kind of associated with us securing our seed route of funding. I've really been able to put so much responsibility in her hands, and it's gone wonderfully. This would be no surprise. Dana's always been such a incredibly capable person to work with in every venue where she tries something. Oh, competent, or outstanding. I mean, it's really been wonderful. So over time, you can ramp up and ramp down how much burden you're taking on as an academic founder. And I'm still very much involved, very much invested, but it's been a delight because now my role is like, okay, what's the thing that only Ross can help with? And then I put all my energy into that. And that for me feels much more rewarding, fruitful, and enjoyable than, oh my gosh, I have to figure out how to get a tax ID number and via some bureaucratic web portal. Yeah, I
Duns and
don't want to
Bradstreet.
be I don't want to be dealing with that stuff. So yeah, it's incredibly flexible in how things get done. They just have to get done. And I guess I'll just tag this with one more little idea. Early on, most scientists would go looking for a protocol. Oh, where's the protocol to start a new company? And people give you an outline, but there are big chunks of that protocol that are kind of like, just figure it out. One of them is like cap table. No one's going to give you the blueprint for a cap table. You just got to do something. And that was a strange experience. Usually there's some precedent, or in a situation where there's so much precedent, it kind of gets condensed into some how-to. That doesn't exist for certain parts of the startup journey. And so that was a surprising element. But sometimes that flexibility is good, right? You just wing it and move on to the next intellectual challenge and keep going.
Yeah, thank you for that. I asked that question because traditionally speaking, when you've taken on a job as an academic, as a professor, or the route to that, people spend 100% of that time to get a tenure. And I just want to open the eyes to other people going down that route, that if startup is on your agenda, this is a path you can follow. You should follow Ross on his website. I'm sure he'll be happy to talk to you guys. I think that's a nice segue to talk about funding. You mentioned about Skydeck, LSEC, and I see that you've raised over $8 million in your seed round. What has your fundraising experience been like? Talk about the legwork that went into it. It's not magic. I'm sure there was a lot of thought that went into it because you're successful, obviously. I see a lot of companies struggling there. What were the conversations like with the investors?
Yeah, I guess I'll take the first chunk of this and maybe hand it off kind of quickly to Dana. So I was in the driver's seat largely for the pre-seed phase, which is a really interesting time. Ideally, you have some limited amount of cash that can start employing people to be dedicated to this mission. So that was secured really with the support of Skydeck and LSEC. So these are both wonderful things that gave us some cash to start deploying. Even things as simple as like hiring a graphic designer to make our pitch deck look a little bit nicer. It goes a long way. And if you have to invest your own personal funds, it can be a little bit daunting, right? So it takes away the fear of that element. And we were also able to work with a CEO that was really driving the roadshow and the interactions with investors. So this is somebody that was kind of launching the company before Dana joined up, Janine Winslow, who was totally instrumental in kind of helping me learn the ropes. She was able to mentor Dana to some extent. And yeah, she was a wonderful partner in those early days when, I couldn't fill that cup all the way. So yeah, huge credit to her. And she was supported by this pre-seed funding. And that makes a huge difference because it's not just me anymore. And I also would like to give some credit to Malu Kanaswamy, who was a nights and weekends business student that was my partner in the Skydeck program. And so she was able to contribute a lot as well. And this kind of allowed us to get to this position where we could speak to people interested in investing in biotech. We had a reasonable pitch deck. We had our vision that was becoming more and more crystallized for what Editpep was going to be. And there's this common phrase you might hear that you'll hear 99 no's before you hear one yes. Yeah. And I looked at some point at our list, our kind of digital Rolodex. Who are all the investors we've talked to? And it's true. You end up talking to 100 people in that course. And I think it was over the course of a year. A lot of those people are just like a total mismatch. I guess one piece of advice I would have for anyone setting out on this journey is spend a little bit of extra time just to avoid that 30-minute or 60-minute conversation with someone who is not going to provide you advice that is helpful, and they're not going to provide you with funding. Mm-hmm. Some of those conversations I had, in retrospect, I could have just skipped them, and I didn't really learn that much, even from hearing the no. So if you're limited on time, I think I could have applied a little bit of a stronger filter. So yeah, there were a lot of productive conversations. Some of those moved towards term sheet, and that was encouraging. But at that time, we got in touch with the organization that ultimately did provide our seed round. And I guess I should hand it over to Dana to talk through that unorthodox, but really, really wonderful opportunity that presented itself.
Yeah. So as Ross said, we got connected with this potential funder. They're called Linden Light Collective. And they are an organization that is dedicated to funding and developing novel therapies for pediatric brain cancer. And Ross took the initial meeting with them, connected via Jennifer Doudna. And I would say that this really is credit to Ross for that open mind. I feel like what he just said, where he could have had a more stringent filter for first conversations, actually would have gone counter to the opportunity that actually came to be very fruitful. So I would say Ross's open-mindedness is really central to the serendipitous interactions that we ended up having with this organization. Because initially, brain cancer wasn't what we had envisioned for one of our first indications. And it was through Ross's conversations with the scientists at this organization that we realized that our platform and our approach really does have applicability in certain cases of brain cancer. And that was a pivot for us because we initially were only focused on treating genetic diseases like epilepsy. And so what ended up happening is through those conversations, we realized that there's another opportunity to treat another disease with a lot of unmet need. And so we pivoted a bit, but it's more like an expansion, not so much pivot, because this organization, Linden Light, has funded our seed round to establish a lot of general platform development that will be applicable for all sorts of diseases of the central nervous system, as well as our original planned pipeline targeting Dravet syndrome, as well as a program looking at developing a treatment for pediatric brain cancer. And so that was extremely exciting. And we are just thrilled to have this opportunity that has been given to us via Linden Light Collective, because of course, it's not just the capital invested, it's also sort of this partnership with a funder that really wants to make a therapeutic. And that to us was the most important thing. After having so many conversations with so many different funders, I think who you get funding from does shape a lot of what directions you go in. And although perhaps going with a non-institutional sort of VC, the sort of standard path, presents some unknowns, we were really excited about the opportunities to really focus on the potential upsides of partnering with an organization that has patients as their absolute focus. And so we are working hard and partnering with them, making progress on that front. And so, so we are 5 months, I think, into our about 2.5 years of seed funding. And it's going great.
This is a fascinating story. This is something repeating in some of my podcast guests. They talk about how patient advocacy groups are funding startups. And I think there's a mission there. I think that's very complementary. I'm glad Linden Light came through for you guys. Before I move on, Ross, like you mentioned about filters, you talked about 100 investors. Like if there are 1 or 2 filters that you would like people to keep it in the back of the pocket, what would they be?
First, I want to comment on the Linden Light conversation because, Dana, you do bring up a good point. Like I could have potentially filtered that out. And it did actually hit when I was pretty deep in filling that list. I was probably at 80, 90 conversations. I was probably getting pretty exhausted with it. And the reason that they made it through is because they are patient-centric, right? So my goal was really to talk to these people because their mission is not laser focused on making money. They really, really want to help people. And I will never turn down that kind of meeting. And I guess this kind of comes back to one of the bigger picture things that relates to the IGI. Aside from trying to generate transformational technology, we have a lot of emphasis here on the ethics and advocacy and accessibility that is pertinent for the CRISPR therapeutic space. So that's something I think about very, very often is how to make these things more accessible. And you can change regulatory strategy, you can change financing or funding strategy, but I do want to say it's very motivating to do the work that we do because that delivery barrier is a technical barrier that keeps these drugs away from patients, right? So we know that this potential is immense. We just want to kind of unlock that potential. And that's a very motivating thing to do, both directly through technology and indirectly through other things that the IGI is focused on, like thinking about patient advocacy and pricing and all this kind of stuff. So back to your question about what filters to apply. It's really basic stuff, and it's almost embarrassing that I didn't realize this earlier in the journey, but certain firms are only going to talk to you regarding your Series A. And when they talk to you, if you're seeking seed funding, they're doing kind of some free consulting to understand what's out there, what the landscape looks like. And it can be good to build that connection. But if somebody says it's too early, either your technology is not a good fit for them or you're just too young and unproven. And so there were some instances where I'd kind of keep knocking on the same door or the same type of door and getting the same answer. And eventually I figured out like, okay, there's this syntax that I had not been interpreting properly. There are these rules that you can pick up on regarding who should be talking to whom and when. And so it's just really basic startup 101 stuff that maybe for some people it goes without saying, but if you look at their portfolio and all the companies have one flavor and it's very different from the flavor of your technology, it's going to be an uphill battle in many cases. But you never know for sure. So if you have someone with the bandwidth to have 200, 300 conversations, that's great if it's not costing you anything. But for the potentially spread thin academic co-founder, you do have to make some strategic decisions about how you spend your time. And I guess I'll also say like 30-minute meeting instead of a 60-minute meeting, that can make a big dent too. So if it's a maybe in terms of match, maybe just keep it brief, right?
Yeah. No, I think this is the common observation for first-time founders, actually. There's that hope that you might get some money from somewhere. You don't know who that one investor is going to be. That's the hope that drives that conversation. But I think it can improve both for the investors and for the founders. Let's shift the gear slightly towards the market. Okay. Alnylam Pharmaceuticals came up to my mind as I was thinking about your conversation. It started in 2002 with a bold vision to create RNAi therapeutics, but they had to crack tough delivery challenges like the RNA to the liver using lipid nanoparticles. Editpep is tackling a similar hurdle with PERC platform to deliver CRISPR to the brain. Are there any lessons that you've taken from them? This is for other people in the space as well, because
Absolutely.
It's a long journey.
Yeah, absolutely. There's so many interesting parallels between the entire field of RNA silencing, oligotherapeutics, and CRISPR. So just a cute little anecdote is that both Ross and I started in RNA silencing. That's where our training is from, that entire field. Before we started studying CRISPR, we were studying the RNA silencing pathway. And that was true, of course, of Jennifer Doudna as well. She was the leader in that field before she was the leader of the CRISPR field. And there's a lot of parallels, as I sort of said when we started our conversation. siRNAs are these large negatively charged molecules. They're also hard to deliver. And really, the oligo field helped pave the way for these learnings around how to deliver these molecules, what was required, what wasn't working. So I would say that Ross and I's entire training is building off of the RNA silencing field, but also the entire CRISPR field, maybe even larger than that. All the gene therapy field is also building off of that knowledge gained from companies like Alnylam. Not specifically, because you basically only learn bits and pieces from what they've learned, but I would say a lot of the lessons are similar. And so I think the thing Ross and I always come back to is that when you have a therapeutic hypothesis, like an siRNA can knock down a gene, CRISPR can target a gene. If you're thinking about it just like a molecular biologist would, or a geneticist, the potential is there. But what we feel has been missing from the field is really a focus on solving the tangible delivery challenges and not maybe just repurposing the delivery technologies that already existed in other fields. So, I think especially in the CRISPR field, we feel that people have been focused on other aspects, really thinking that delivery is, in the initial phase. I think now everybody is very aware that delivery is the main challenge. So it's sort of an easy pitch for us at this point because people get that that's a challenge. But in the earlier days, people were really focused on, making different enzymes and all sorts of other aspects that are valuable as well. But ultimately, it doesn't matter what nuclease you're using if the delivery is not practical, it's not efficient, it's maybe immunogenic. All of these aspects really trump all the others.
Yeah, great. And these challenges are immense, especially with the delivery, right? It took about
Right.
16 years for Alnylam for the
Right.
journey with
Right.
Onpattro.
And that's what I was going to mention is that arc, because I think oligos are now having their moment where they're on the rise. And I feel like a lot of that, Alnylam really paved the way, and they had to do a lot of hard work and creative work, kind of sticking it out in that field. I really admire their path. And that also gives me hope, because also a little bit of a downturn, or people are feeling a little pessimistic about CRISPR right now. But Ross and I don't feel that pessimism whatsoever, because we really feel like the reason people are pessimistic is because the delivery challenge wasn't fully paid attention to the extent that it needed to. So with our focus on solving that problem, we remain extremely optimistic that our vision will be realized in a therapeutic.
Yeah, thanks for being persistent on that problem, right? Drug development, by all means, is still hard. It's a long journey. Is there a framework in your mind about this journey? I really wish and hope it won't take 16 years or whatever for you guys, but I want to see how you think about it. It'll also help the audience to jump into this space. How do you see this problem,
One thing I really want to shout out is this program that the NIH has been running for the last 6 years or so, or maybe 7 by now. It's called the Somatic Cell Genome Editing Consortium. So this is something that was conceived right as CRISPR started to move towards the clinic. I think leadership, both in academia and in the federal government, saw this massive potential, but they really respected the challenge represented by delivery. So what SCGE Consortium did was put a challenge out there to the researchers in the CRISPR space and the delivery space. And they essentially said, look, we want you to figure out a way to use non-viral delivery to reach organs other than the eye and the liver. They kind of saw that the eye and the liver were going to be tractable, but everything else in vivo was sort of off limits at the time. And they funded 20 different academic grants. And I was actually a collaborator as part of 2 of these efforts. So I was a kind of leader of one and then a major collaborator in another one. And that investment has borne fruit via Editpep. Dana's work on ex vivo peptide-mediated delivery, that was one program, actually ended up launching our other program, in vivo brain delivery, to the next level. So it actually took 2 grants to get us to this one technology, and it was something no one really saw coming, but it's been a remarkable breakthrough. That time frame, just saying take 5 years, it's the span of one of these NIH grants, saying take 5 years and develop some technology with this explicit focus, that's something that doesn't typically happen in industry. And in academia, people seldom have access to the resources necessary to answer those questions in a meaningful way. So one of the coolest things NIH did was set up a testing center, or multiple testing centers actually, dedicated to getting reagents into large animals. This is something academics almost never have access to. And through this consortium, people are doing genome editing in non-human primates and in pigs, which is just a total game changer in de-risking this leap from mouse to larger animal, which is totally essential for clinical translation. So the NIH was the key enabler of this really focused development of technologies that will solve real world problems, but it's going to take more than 2 or 3 years, which tends to be the sort of level of window that you might have with traditional venture funding. And so I think this has been a major disconnect between the unmet need over here, which is causing all these headaches in the clinic, and then an actual scientist kind of lacking the focus and maybe lacking the longer term support to really, really solve these challenges. So the Alnylam example is great. I mean, all the oligotherapeutics, none of that stuff was solved in just 2 or 3 years, right? It's a kind of long journey, and it's really important to find a match between some kind of funding support and the people with the capacity to move things in the right direction. So I feel very, very privileged to be at the Innovative Genomics Institute, where we have the intentionality to be pointing in this meaningful direction for enabling CRISPR therapeutics. And also, fortunate that the NIH was a major supporter of this work, both directly through funding and also indirectly through setting up these testing centers, which were massively enabling for the work that we're doing and continue to do.
Yeah, I think I want to emphasize the role that federal government and the NIH has played in fundamental research and translation of that research. I really hope people are listening and that continue to support such programs so we can get better drugs. Let's slightly shift the gears. I want to get into some bigger questions. What is the most counterintuitive decision you've made in building Editpep that has turned out to be a crucial step for your success so far? Anything that people don't get?
I would say one of the big leaps that we had to make is overcoming the uncertainty associated with receiving seed support from a non-traditional investor. I talked about how there's not really a blueprint, but there are some elements that are well defined in the protocol of taking technology into a startup. And one of the things that is almost treated as law is that you will take traditional
venture money,
venture funds.
Yeah.
Right?
The angels and the ventures, yeah.
Exactly. And we reached this turning point where we sort of had some term sheet options over here from traditional VC, and then we had this non-traditional investor. And we ended up going with a non-traditional investor. And a lot of people really would take pause at that decision. And we have a network of trusted advisors. And it was something we took very seriously, deviating from the norm in this way. At the time, it was kind of a leap of faith. And you never know how the landing is going to go. We're still kind of mid-leap, I would say, in some regards. But as far as the time that has passed since we made that decision, I think that we have absolutely no regrets. You know, we've been delighted with this path because our funders have been extremely supportive. We've seen other companies just vanish, basically, because the investor optimism disappears. Again, because the focus is a little bit different with traditional VC, right? If they don't see return on investment within that time horizon, sometimes the plug gets pulled. So we feel really good about our decision to work with an organization that, of course, wants to get return on investment, but also is prioritizing patient care as a major pillar of what they're interested in. So I would say that is something that was counterintuitive and really not on our roadmap as we imagined it. However, I will credit this very podcast for letting my mind be more open than it would have been, because some really important companies from our ecosystem, namely Caribou and 4DMT, I think when I heard from the founders on this podcast, they talked about the non-traditional investment that they started with. And maybe it's not a more patient-centric group, but they really sent a strong message that when you have something with any sort of platform capacity, it can actually be valuable to go with a non-traditional investor, because just like we were talking about with the journey of oligotherapies and this path towards getting CRISPR into a functional non-viral state, it can take a little bit more time just to solve the problems. And the traditional VC might not give you the horizon that is a good fit for the technology.
That's a great point. And thank you for the shout out. I think an extension of that counterintuitive step you've taken is it also relates back to the investors. Like, are there any second order consequences of your technology that most people are not seeing yet? If an investor knew about this, they would jump and like hand you $100 million check.
Well, yeah, there's a lot of opportunity. And at this stage, we're just scratching the surface, because I think in your seed stage, you're focused on getting a platform technology really de-risked and set up, poised, I guess it would be the word, poised to then expand into several different therapeutic areas. And so that's what we're focused on now. And so I would say that the opportunity that you're mentioning is very much there, and we are getting set up to be able to showcase the possibilities. I think there's other tissues that we could be tackling. There's systemic delivery is sort of our holy grail that we're trying to develop the technology for, because we have a platform technology that we really believe in. There's going to be many opportunities that expands past what we're currently focused on, but we have to show that it works, and it works in a couple different settings, and then we can really take off from there.
Great. I just wanted to jump into the personal side. Startups are really hard. Like, I'm a founder, I know the pain. If you have a personal startup survival kit, if you have friends, family, tools, technologies, I'll probably plug in one thing, like because you mentioned caribou, Rachel Horowitz, when she was on my podcast, she was talking about how she neglected her health and how she goes on runs and stuff like that. Like it's important to be conscious that you'll take a hit, you'll damage your health. And that's why I'm highlighting this point here.
Yeah, I don't want to endorse any of that stuff. I mean, I definitely have lost so much sleep because the challenges of my typical day job are so great. Developing technology is not trivial. It really, really takes a team. And gosh, I wish I had more time. I mean, I don't have any kids right now. I don't know how Dana manages it. So big respect to you and your family, Dana, for doing what you guys do. No, I don't have a survival kit. I need a survival kit. I need something.
Yeah, it's a challenge. What's my survival kit? My survival kit is a good sense of humor. I think that goes a long way. I would say Ross is the same. It's stressful. I think Ross and I's good relationship helps a lot. I can't even imagine doing that if that was fraught in this whole process. I can't imagine the sort of stress that it would be if we had a fraught relationship. Having a good team, our early team members are so fantastic that it's just an absolute delight to be working with them. But on a personal level, yeah, lots of support. You know, I have 2 young kids, and most people are very surprised at that. And that is definitely a challenge. I think it just means that you have to adapt and you have to get support in every aspect. It's just a lot to juggle. But if you sort of keep as your guiding principle that it's possible and you just have to find the solution, and you might not get every day correct. Some balls are going to drop, but you just keep going and keep pushing through. That's how we're doing it every day.
It's hard. You're relatively new in this startup space. I've been doing this for 12 years now. Left my postdoc in 2013 and I'm running one right now. And I think what really helped me was friends.
Yes.
Friends who are not in the startup space.
Yes.
You just go, like you said, a good sense of humor, laughter, have a drink, eat, and just walk away. I think that's been a constant in my life, and having that spousal support, right? We can't do any of these without that. Well, I hope you stay healthy. That's my message here.
Yeah.
Be happy. Dana, about the future goals, what are the next big milestones for Editpep? And what do you hope to achieve in the coming years? And if there is a specific ask from the audience.
Yeah, we are de-risking the technology for direct brain delivery. And our key milestones are around showing that we can edit enough of brain tissue to have therapeutic benefit for several different diseases. And so we are doing that both in mice and in larger animals such as pigs. So getting the platform to that stage where we can show the potential, even just the first step, which is in reporter models, really proves to people that we can deliver on this promise of editing enough of the brain tissue to have therapeutic benefit, and do it in a way that most people had never dreamed was possible, without a virus, without lipids, with just a protein, a functionalized protein. So that is a massive technical challenge that we are embarking on, but we have, a lot of promising data so far that we're going to be able to achieve that.
Great. Well, thank you for sharing that. I've decided to frame a new closing question for the podcast. You'll be my first guest to answer this question. So some context to this is, I'm sure you heard about Steve Jobs' Stanford speech about connecting the dots, how life's random moments make sense only in the hindsight. For our listeners, many PhDs and postdocs, right? This idea is powerful as they consider startups. I want you to reflect on your path to Editpep. Ross, maybe you can start off by connecting dots that led you here.
Yeah, happy to. This one might seem a little bit odd, but it's really relevant to the stuff that we think about, like some of the most challenging things that we tackle with my lab's technology, with the Editpep technology. It goes all the way back to when I was probably 9 or 10. I was in grade school, and they asked us these like fun science brain teaser questions. And one of them was, if you shampoo your hair, rinse, and repeat, why is it that there are more bubbles the second time you apply shampoo? And I was a kid, and they say, go home, think about it, and tell us the answer tomorrow. And I was like, what? And so I went and I asked my dad, who's like a medical doctor, and he gave me some sort of like hand wavy answer about grease or something. And it was like only like 10 or 15 years later, when I learned all of the thermodynamics and the biophysical chemistry, that I could really understand the spirit of that question. And it's all about hydrophobicity and the behavior of detergents and all this stuff. And that question kind of haunted me because this thing that can seem like it has a simple answer when you just look at it on the surface. Okay, oil absorbs shampoo, whatever. When you really think about the mechanism of this at the kind of molecular level, it can be such a fascinating and complicated thing. And the way that this connects to Editpep is that the challenge that we're trying to do is get across this cell membrane. That's really the critical moment of delivery. When do you get your enzyme from this side of the wall to that side of the wall? And getting across relies on these peptides that we use, and they have a little bit of the same properties as a detergent, same properties as a shampoo. And so we actually think about this and keeping things soluble and not too greasy. It's really a big part of the technical challenge. And there's almost no papers that really tell you how to think about this stuff. So again, a thing that I really love about working with Dana is that we have this background in biophysics, so we can kind of tap into some of this thinking that maybe a geneticist would just never think about. So yeah, for me, it's really things that have simple explanations on the surface, and there's a lot of complicated stuff going on underneath. And sometimes you have to think a little bit more creatively, maybe more like an artist than a scientist, but it can be fruitful. And I guess since I invoked the peptides, I just have to say the cutest thing about the Editpep journey is that I'm going to talk about this company. We're using editing, using peptides. So Editpep, okay, put it together. And I was arguing with my partner. She said, no, it should be pep edit. It should be pep edit. You don't want Editpep. It doesn't have the right vibe. But then I realized that Editpep is peptide spelled backwards. And then history was made. Voila. It was locked in. We couldn't go back. So yeah, we need to find a tagline that latches onto this, but that's for the future. So more brainstorming between me and Dana. Anyway, thanks so much for having us. I'll turn it over to Dana.
Yeah, Dana, how about you? What dots could you connect?
Yeah. I mean, I think I have two answers to this question. It's hard to choose, but one is on the technical side, as Ross is, in terms of what inspired me to go on this journey of the technology. And like I mentioned, we both started in the RNA silencing field. And during my PhD, I was working on developing biotechnology tools using protein that binds siRNA. And it was through that project that got me interested in siRNA delivery. And then I asked the question whether this small protein could deliver the siRNAs into cells. And that is what got me, I tried first just with maybe traditional TAT peptide, and it didn't really work. And then that got me diving into this literature of endosomolytic peptides, which I'm really getting into the weeds now, but that's these class of peptides that really help you. That's really the secret sauce of what we do, where we use these peptides that sort of mimic the viral ability to get into cells and get out of the endolysosomal pathway. And so they're able to burst the endosomes. And that's what lets them transit to the cytoplasm and nucleus. So I got interested in these peptides as a PhD student, and then it was getting connected with Ross. We knew each other actually at that phase, just from conferences in the RNA silencing field. And then when I heard that his lab was going to be dedicated to delivering CRISPR enzymes as proteins, actually as a protein RNA complex, that was exactly what I had been working on in my PhD. So it was almost too good to be true. I was looking at lots of different postdocs in different areas of sort of deep RNA biology, biophysics of RNA, all sorts of things. And then I really got interested in this practical application of CRISPR delivery using the same type of approach that I had been using in my PhD. And so I think that's the biggest dot in terms of, it is serendipitous, that journey, and it certainly wasn't by design. And so it just was a winding path that led me here. So that was on the technical side. And I'll just do one more kind of on a more personal note. I would say that, I'm a first generation college student. My parents were small business people. And I think growing up in that environment is also what teaches you a little bit of the gumption, as I mentioned earlier, to embark on paths that are maybe not as well defined, and to have the ability to take a leap of faith on an idea. And maybe even if your professional journey isn't totally clear as to going to grad school and then going to postdoc and then starting a company, that all felt, even though it's completely different from the environment I grew up in, it felt familiar in that I don't need to have all the answers right now. I can take a leap of something that I'm interested in now and try to see where it takes me and explore from there. And so I credit sort of that environment with the ability to take chances.
This is great. Thank you for sharing that, because I think that's the message I want to send across to the audience. Embrace nonlinear paths. I think it's going to build you resilience and confidence, and recognizing serendipity is hard when you're in the position. I think when you look back, you'll connect the dots there. I hope everybody else can and will. But thank you so much, Dana, Ross, for sharing the story so far. I'll be cheering you from the sidelines here. And I hope to bring you back in 2 or 3 years to tell the next part of the story. And if there's a big exit, we'll celebrate and do a bigger story then. But good luck with everything.
Thank you. It's been a pleasure.
Thank you so much. Yeah, thank you so much for the opportunity to be here and to chat with you.
Absolutely.
Thank you for listening to this episode of the Lab to Startup podcast. You can find links to the resources and programs mentioned in these episodes, connect with Naresh, or subscribe to this show at labtostartup.com.